Hi, this is Mr. C Biology, and today we're looking at protein synthesis. So here we have the nucleus of a cell. Inside the nucleus we find DNA. DNA contains a code to make proteins. However, it has a problem, and we'll get to that in a second. Here we see the nucleus in the cytoplasm of a typical animal cell. Let's have a closer look. We've just shown DNA as a simple line for the moment. The problem that it has is that it can't get out through the nuclear pores. It needs to get its code to the ribosomes in the cytoplasm. You can think of these ribosomes as protein factories, but they can't make proteins without the code from the DNA. DNA is usually double-stranded and has a code that is made of 4 chemical bases. These are called A, T, C, and G for short. These letters are the code that will make proteins in the ribosome. We also have a single strand of RNA. It has the same 4 bases except for the T. In RNA, thymine, the T, is replaced by the letter U for uracil. You should also note that in RNA, G always matches up with the C on the DNA. U matches with A, and A matches up with T. RNA and DNA are both made up of units called nucleotides. 3 of the nucleotides in a row are called a triplet or a codon. Looking closer at a nucleotide, we can see that it is made up of 3 main parts. It is made up of a base, a sugar, and a phosphate. The base can be A, C, T, G, or U. The sugar is ribose. The sugar and the phosphate actually form the side of the DNA or RNA molecule. It's like a rung on a ladder, and the base is like a step on the ladder. Let's get back to the DNA in the nucleus. It can't get out of the nucleus, so it gets RNA to make a complementary strand. As you can see, the bases match the code of the DNA. C matches G, G matches C, A matches T, and U matches A, etc. This means that the code has been copied into the RNA molecule. We call this transcription, as the DNA code has been written across or transcribed to the RNA. This RNA is now carrying the code from the DNA. This type of RNA is called mRNA. The M stands for messenger, as it carries the code like a message out through the nuclear pores and into the cytoplasm and then to the ribosomes. The ribosomes are also made of a type of RNA, and this type is called rRNA, and the R stands for ribosomal. There is one more type of RNA we need to know, and we'll get to that shortly. The mRNA moves out of the nucleus and to the ribosomes. The ribosomes are able to scan the code and make proteins. The ribosome reads the messenger RNA code in 3s, or codons. The first code to be read here will be AUG. Also found in the cytoplasm near the ribosomes are the 3rd type of RNA, called tRNA. The T stands for transfer, and is called this as its job is to transfer amino acids to the ribosome when they are called. Each codon on the mRNA is read by the ribosome. The example shows codon AUG being read and then matched with the anticodon of UAC in the tRNA. The tRNA moves to the ribosome and joins the codon and the anticodon together. This particular tRNA has no amino acids attached. This is because it's a special type of codon called a start codon. It tells the ribosome that it's going to start making a protein. Once production has started, the next codon is read by the ribosome. The UAG on the mRNA is read, and then the matching anticodon is called from the tRNA. The matching AUC moves to the ribosome and brings the first amino acid to start the protein synthesis. This process is known as translation. as the mRNA code will be translated or turned into a string of amino acids. The next mRNA codon of CAU will attract the tRNA with GUA anticodon, and this carries the green amino acid. There are about 20 different amino acids, each with matching anticodons. The amino acids form a peptide bond, which holds them together when the tRNA drops away. The process of reading the codons, then calling the anticodons, and bringing over the correct amino acids continues. As long as the DNA code is correct, then the mRNA code will be correct also. This means amino acids are added in the correct sequence, and this makes the correct protein. Finally, after at least 200 amino acids, the final codon on the mRNA is read by the ribosome. This code is UAA. This is known as the stop codon, and it tells the ribosome that the protein synthesis is complete. The protein is finally released into the cytoplasm. The protein must be folded in a certain way in order for it to work properly. The mRNA code can be read over and over again by many ribosomes, and the proteins will be made throughout the cytoplasm. The proteins are then used in the cell and in the body for growth and repair, or used as enzymes to speed up chemical reactions. I hope this video helped you with this tricky topic. You may need to watch it a few times as there's quite a lot going on. Please don't forget to like, subscribe, and share if you found it useful. Thanks, and see you in the next video.